Hello viewers, in this video we are going to learn about bioremediation of xenobiotics. First of all, let us know what is bioremediation. Bioremediation is a technology that utilizes the metabolic potential of microorganisms to clean up contaminated environments. So here we are cleaning up contaminated environments with the help of microorganisms. One important characteristic of bioremediation is that it is carried out in non-sterile open environments that contain a variety of organisms. So already there are a variety of organisms and the conditions of bioremediations are not sterile. So we carry out the bioremediation along with the natural microbiota of in the contaminants. Okay. Now what are xenobiotics? The term xenobiotics is used for pollutants such as dioxins, polychlorinated biphenyls, and their effect on the biota. Because xenobiotics are substrates foreign to an entire biological system, that is, artificial substances, which did not exist in nature before their synthesis by humans. So, xenobiotics are foreign to life. So, they are only present once the humans have created them or synthesized them. And the components of the term are derived from the Greek words xenos and bios, which means foreigner or stranger, and to life. Bios means life, so foreign to life, and then suffix is used as tic. Okay, so xenobiotic. Now, xenobiotics may be grouped as carcinogens, drugs, environmental pollutants, food additives, hydrocarbons, and pesticides. Right, so they can fall into different categories, but they are no new to life or foreign to life. Sometimes xenobiotics can also be those substances or compounds which are present in nature, but when human due to human activity or anthropogenic activities, they pile up in so high amounts that cannot be handled naturally by microbes. Okay, so then those can also fall into the xenobiotic category. As I told you that xenobiotic compounds are man-made chemicals that are present in environment at unnaturally high concentrations, and xenobiotic compounds are either not produced naturally or are produced at much lower concentrations than by man. Then there is a principle given by Alexander it was given in 1965, which is principle of microbial infallibility. It means that microorganisms or principle of microbial infallibility says that microorganisms have all the capability of degrading all the naturally occurring compounds. So whatever is present in nature can be degraded by microorganisms according to this principle, but which is not present in nature, which are not they are present naturally in environment, so they might not be subjected to degradation by microorganisms because they are foreign to life. The recalcitrant xenobiotic compounds means compounds that resist biodegradation and thereby persist in nature or persist in environment. These compounds can be grouped into 6 categories or 6 types: halocarbons, polychlorinated biphenyls, synthetic polymers, alkyl benzyl sulfonates, oil mixtures, and all the other chemicals which doesn't fall in these 5 categories. So halocarbons, polychlorinated biphenyls, synthetic polymers, alkyl benzyl sulfonates, oil mixtures, and other chemicals which are not falling into above 5 categories. Now, xenobiotic compounds, as I told you, can be pollutants, can be pesticides, can be synthetic azotides, can be polycyclic hydrocarbons, and even polyaromatic chlorinated and nitroaromatic compounds. Now, all of these compounds, xenobiotic compounds, show some or exhibit some properties like their molecular weights are very high. They may contain condensed benzene and pyridine rings. They may be polycyclic compounds. They exhibit low solubility. They have higher number of substituted nitrogen atoms, and they also have quaternary carbons. Okay, so these are some properties that make them very recalcitrant or non-biodegradable by microorganisms. So first of all, let's see what halocarbons are there, which are xenobiotic. These compounds contain different numbers of halogens that can be chlorine, bromine, fluorine, iodine atoms in place of hydrogen atom. So hydrocarbons where hydrogen atoms are replaced by halides, okay, halogen atoms, are known as halocarbons. They are used as solvents like chloroform and CHCl3, right, or as propellants in spray cans of cosmetics, paints, etc., and also in condenser units of cooling systems like freons. Different freons are used in cooling systems, and also as insecticides like DDT, BHC, Linden, etc., and also herbicides like Dalapon, 2,4-D, 2,5-D. All these are halocarbons. The C1C2 haloalkanes like chloroform, freon, etc. are volatile and escape into atmosphere where they destroy protective ozone layer. We know that these halocarbons are known to destroy our ozone layer, and that leads to increased UV radiation coming onto the Earth's surface. Right? Pesticides, including herbicides, fungicides, and insecticides, are applied to crops from where they leach into water bodies. Right. So once they are applied, all of it will not be used. Rather, some of it or most of it will leach out in the water bodies, and many of them are subjected to biomagnification. I'm sure that you are aware of what biomagnification is. Biomagnification is increasing concentration of substances, including toxic materials or xenobiotic, in the tissues of tolerant organisms at successively higher levels in the food chain. Right. So top uh, carnivores will be having the highest concentration of these toxic compounds. Right. So next xenobiotic compound class is polychlorinated biphenyls (PCBs). Right. These compounds have two covalently linked benzene rings. Right. These two benzene rings are covalently linked, and these are having halogen substituting the hydrogen. Right. PCBs are used as plasticizers, insulators, coolants, in transformants, insulator, coolants in transformers, and also as heat exchange fluids. Right, they are both biologically and chemically inert to various degrees, which increases with the number of the chlorine atom present in the molecule. So as the number of the chlorine atoms increases, its inertness, biological and chemical inertness will increase. The recalcitrant nature of the halocarbons as well as polychlorinated biphenyls is due to their halogenation as well as their cyclic structures. Right, so they are halogenated as well as cyclic, so that makes them very recalcitrant, very tough to be degraded biologically. Now, synthetic polymers are the compounds produced as plastics like polyethylene, polystyrene, PVC, nylon. These are used as garments, wrapping materials, etc. These are recalcitrant because they are insoluble in water and they're large molecular. Their size is really large, molecular size is really large. So basically, these two are the reasons behind their recalcitrant nature. Right. And alkyl benzyl sulfonate, as you can see, have a benzyl group and alkyl group. Right. These are surface active molecules superior to soaps because they have a hydrophilic and hydrophobic region. Right. Amphipathic molecules they are. Sulfonate group present at one end resists microbial degradation, while the other end, which is non-polar alkyl end, becomes recalcitrant because when it is really branched. Okay, so the higher the branching, higher the resistance, and higher the recalcitrance. And at present, alkyl benzyl sulfonates having non-branched alkyl ends are used, and these are basically degraded by beta oxidation from their alkyl end. If they are non-branched or unbranched, they will be degraded at starting from their alkyl end. Oil mixtures, oil is a natural product. We all know that oil is produced in plants and microbes, right? So this is a natural product, and it has many components, and is biodegradable. The different components may be degraded at different rates, but overall it is biodegradable. Biodegradation is able to handle small oil seepages, like small spill or very small amount of oil can be handled through biodegradation. But when large spills occur, the problem of pollution becomes acute, and then it becomes really tough to biodegrade that or tackle that situation. Oil is recalcitrant mainly because of its insolubility in water and due to the toxicity of some of its components. Other xenobiotic compounds, a number of pesticides are based on aliphatic cyclic ring structures containing substitution of nitro, sulfonate, methoxy, amino, carbonyl groups, right? And in addition, they also contain halogens. These substitutions make these compounds really recalcitrant. Now, what are the effects of xenobiotics? Xenobiotics affect health, soil, and water. They can be mutagenic, teratogenic, carcinogenic, or allergenic, right? So they do make an impact on health, and the soil parameters are can also be altered. These xenobiotic can be assimilated by plants, and they can then make their way to food web, right? And they can be transported with soil eroding particles to water, ultimately polluting the water bodies, and then accumulation in animal food chain is also always a possibility. Then sedimentation of hydrophobic pollutants in water pollute the water, right? And there can be changes in physicochemical characteristics of water because of the occurrence or presence of xenobiotic compounds, right? Now, factors that affect biodegradation of xenobiotics: how biodegradable they are, how many sulfonates are there, how many alkyl groups are there, how many benzyl groups, how much branching is there, how many halogen elements are there, right? So how that all will decide the biodegradability. Okay, so what is the biodegradability of the compound, right? This is an important factor. And what is the mineralization potential? Mineralization means how they can be degraded to their elements, right? And can be reabsorbed in the system. Now, availability of carbon and energy sources to microbes, right? If energy and carbon is there, sufficient inducers are there to activate genes required for biodegradation of xenobiotics, and that will also affect degradation of xenobiotics. And availability of other nutrients like nitrogen and phosphorus also affects xenobiotic biodegradation. And then extent and distribution of contaminants. How many contaminants are there? What is the amount of the contaminants? Whether they are more recalcitrant or less recalcitrant, or whether they can be used as carbon energy source or not. And then availability of electron acceptors and redox. What are the redox conditions, right? What are the acceptors available? What are the redox conditions? Prevailing redox conditions will also drastically affect the degradation of the xenobiotic compounds. Then sufficient number of degraders should also be there in non-sterile environmental conditions. Either we can exogenously add these degraders, or if they are naturally present, then the sufficient number of degraders should be there. So all these parameters affect degradation of xenobiotic compounds. Now you can see in this table there are different bacteria that degrades many target compounds, right? Like Mycobacterium species are able to degrade endosulfane compounds. Then Dihalospirillum species are capable of have been reported to degrade DDT. Then Dichloromonas species has been reported to degrade benzene. Like other microorganisms have been reported to degrade RDX, petroleum products, azotides, etc. Right, so we have ample a wealth of microorganisms capable of degrading xenobiotic compounds. Right, and there are different pathways operating for degradation of these compounds. Like there are aerobic mechanism, anaerobic mechanisms, or even co-metabolism exist, which are employed or used by microorganisms for degrading xenobiotic compounds. Say, for example, if a polycyclic compound is degraded through aerobic pathway, it will first undergo oxygenation, and then some oxygen atom will be introduced in the system or the molecule. Say, for example, polycyclic compound get oxidized into a dihydroxyaromatic compound. Then further, the ring cleavage may occur, and it can become a carboxylic acid uh, or a cycle intermediate. Right, this way an aerobic pathway will operate. The reductive decolorization of sulfonated azotides. Reductive decolorization of sulfonated azotides are efficiently accomplished, and there are n number of organisms that can carry out that, like Pseudomonas, Sphingommonas, Xanthomonas, etc. Right. So it's really well documented that these compounds can be degraded by microorganisms. Now this is an example of aerobic degradation of benzene. You see here, it can be degraded in two different ways. Say for molecular oxygen, in the presence of molecular oxygen. So it's an aerobic process. Benzene dioxygenase oxidizes benzene into dihydroxy cyclohexane hexadiene. Right? Two double bonds are there, so diene. Now then it is further uh, isomerized into catechol. It's an, again an aromatic compound, but with higher number of oxygen elements. Now another pathway is that formation of an epoxide will occur with the help of another monooxygenase, another oxygenase. It is a benzene dioxygenase and this is benzene monooxygenase. So with the help of monooxygenase, an intermediate is formed, that is benzene epoxide. So an epoxy compound intermediate is formed that ultimately is hydrolyzed into the same, like dihydroxy cyclohexadiene. Right. Further degradation or conversion will be in the form of catechol. Right. So catechol is the product, final product, which will further be degraded by other enzymatic reactions. Now, if we take catechol to acetyl coenzyme, which is really assimilated in the pathway, the different pathways, metabolic pathways, right? So, catechol is subjected to degradation with the help of 1,2-dioxygenase, and then muconate is formed. This muconate undergoes cyclization to form muconolactone, a cyclic lactone, right? And then it's undergo isomerization and forms enol lactone, right? And then oxidize, this ring opening occurs and an oxoadipate is formed. Similarly, isomer of catechol can also be uh, subjected to oxidation degradation, and ultimately again the product will be 3-oxodiadipate enol lactone, right? Which ultimately will be giving the same product, like 3-oxoadipic acid, which is further metabolized through with the help of acetyl coenzyme, coenzyme moieties. So first we get oxoadipate coenzyme A, and then we get acetyl coenzyme A and succinyl coenzyme A. And you know that these are really popular names, acetyl coenzyme A and succinyl coenzyme A. So these compounds are then channelized through TCA cycle, right? So catechol compounds like catechol and their isomers can ultimately make their way. Their carbon will be recycled to other the more readily assimilable molecules like acetyl coenzyme A and succinyl coenzyme A. Right. Similarly, cyclohexane, which is a non-aromatic cyclic compound, or so this also undergo oxygenase action. This is acted upon by oxygenase, and cyclic alcohol is produced, and then further oxygenation converts it into a cyclic ketone. Then further is oxidized. Then the chain opening results in formation of an acid. Once this alkyl, this acid is formed, acid alcohol, this can be metabolized through beta oxidation pathway, and acetyl moieties will be released for assimilation, right? So this way, this cyclic hexane compound can also be metabolized. Anaerobic pathway includes dechlorination, halorespiration, followed by hydrolysis, nitroreduction, alkylation. These steps are involved in anaerobic pathway. Usually, phthalate compounds degradation is mainly carried out by anaerobic methanogens. Right, so anaerobic methanogens. So these organisms include methanospirillum, methanoseta, and centrophobacter. Okay, these produce upon degradation of phthalate compound, they produce acetates and methane as end products by decarboxylation initially, and then reduction followed by ring cleavage and ultimately by beta oxidation pathway. Right, so acetate and methane are the main end products of phthalate degradation by anaerobic methanogens. Now you should also be familiar with some terms like co-metabolic pathway. What is a co-metabolic pathway? Sometimes microorganisms can degrade these xenobiotic compounds like cyclohexane and other halogenated compounds, but degradation may not serve as a source of energy and carbon for them. So they need to depend on some other substrate for deriving energy and carbon, right? So degradation of xenobiotic compounds here in this particular case will depend on the presence and metabolism of a suitable substrate, right, which will act as carbon and energy source. So this kind of a degradation is known as co-metabolic pathway. and the compound that is degraded and is serving as source of energy and carbon is known as co-metabolite, right? So this kind of a pathway where another substrate is used as for a source of carbon and energy, and it is required for co-metabolic degradation of these xenobiotic compounds. So this will be called co-metabolic pathway of xenobiotic compound degradation. So here you see the different uh, you know molecules or substrates, co-substrates that can act as co-metabolite like methane, methanol, propane, propylene. All these can act as a co-substrate for degradation of contaminants like TCE, DCE, VCE, etc. Right, and the enzymes that are involved for degradation, because these compounds will be serving as source of energy. So for methane, methanol, compounds like these will methane monooxygenase, methane dehydrogenase, etc. will be required, right? Or similarly, ammonia and nitrates can be used for as co-metabolite for degradation of TC, EDC, etc. TNT, VC, right? And ammonium monooxygenase will be the enzyme that is required for metabolism of these compounds. Then some comparatively recalcitrant compounds like toluene, butane, phenol can also act as co-substrate for degradation of compounds, contaminants like these. For these specific enzymes, oxidase enzymes like toluene, monooxygenase, dioxygenase will be required, and also methanol in anaerobic condition can serve as a co-substrate for metabolism or xenobiotic degradation of PC, TC, DC, etc. using alcohol dehydrogenase having microorganisms, right? So like these substrates like glucose, sulfate, lactate, pyruvate can serve as co-substrate in anaerobic condition for degradation of beta-X, PCE, etc. And the enzymes that are required are dehalogenase and dichloromethane dehalogenase, etc. And many microorganisms are known to possess these enzymes and use these compound or co-substrate and co-metabolites for degradation of many contaminants, right? So it's a really common phenomenon in microbial world to use co-metabolite for degradation of xenobiotic compound. Now another term that we should be familiar with is gratuitous metabolic pathway. What is this? Actually, in contrast to uh, this co-metabolic pathway, in gratuitous metabolic pathway, xenobiotic compound also serve as a source of energy and reducing equivalents, and hence this is known as gratuitous metabolism. In such a metabolism, the necessary enzymes are already induced by another compound, which is actually not serving as a co-metabolite. So both the xenobiotic compound is serving as both contaminant here, which needs to be degraded, as well as the source of energy and reducing equivalents for the survival of the microbe. So this way, gratuitous metabolic pathway works. And always the ultimate fate of biodegradation is that production of simpler compounds like CO2, water molecules, nitrate, nitrites, sulfates, like all these compounds are released upon complete degradation of or complete mineralization of the xenobiotic compounds. In aerobic respiration, we know that the ultimate oxidant or electron acceptor will be oxygen, and in anaerobic respiration, other compounds will serve as electron acceptor, while in fermentation, both organic substrate will serve as electron donor and electron acceptor. Okay, and end products in this case can be organic compounds, carbon dioxide, methane, etc. Right. So I hope that this video is useful for you guys. So we will have another lecture on biodegradation of xenobiotic compounds with some more details. Uh, so thank you for watching this video. Keep coming to my channel. Do subscribe and like these. Thank you. Thank you for watching.